Thank you very much. It is a great honor for me to be here. And can you hear me? No? Okay. So I want to say that it is a great honor for me to be here, and that I want to thank the organizers for allowing me to present our work. In the talk that I am going to give you today, I have divided it into three parts. First, I will give an introduction in which I will talk about Ecuador and frog reproductive modes. I will give you an idea about the biology of 4 different frogs, although this picture just shows 3. Just think that the fourth one is Cenopus. Okay, is that better?
Let me take this and try to fix it. Maybe it's just a battery.
Okay. Thank you. So in the first part, I will explain about the biology of 4 frogs. This image here is from one of the cages in which we keep the frogs. And as you can see, there are 3, but the fourth one is Cenopus, which provides the comparison. And in the second part of the talk, I will do a comparison of frog early development. I work at the Pontifical Catholic University of Ecuador in Quito, and I thought it gives me an opportunity to give you some introduction about the country. Ecuador is a small country located in the northern part of South America, between Colombia and Peru, on the Pacific Ocean. It is crossed by the equator, and that provided the reason for the name of the country. The Galapagos Islands belong to Ecuador. They are also located on the equator and separated from the mainland by 1,000 kilometers. The Andes cross the country from north to south, and that provides a diversity of geographic characteristics which are related to the great biodiversity of Ecuador. Ecuador is one of the most biodiverse countries in the world. I just wrote here that it is the home of 10 to 20% of all terrestrial vertebrates. Those vertebrates are mostly you see, this thing is sort of complicated, but please bear that, okay? So it is the home of 10 to 20% of the terrestrial vertebrates, but those are not the big animals that you see in Africa. They are smaller animals, mostly frogs and reptiles. In fact, look here, Ecuador has 443 species of frogs. Larger numbers of frogs exist in Brazil and Colombia. The 3 countries have the largest biodiversities of frogs in the world. But think of the surface area of Ecuador in comparison with Colombia or with Brazil. So per unit of area, we have a higher biodiversity of frogs. And so I think it is appropriate to talk about frog development. Since everything is being tested here, I'm going to see if I can change the slides this way. Okay, that's the way to go. Okay, so an important thing to see is to talk about frog reproductive modes. When you think of frogs, frogs deposit the eggs in the water. Thank you. Please help. Frog eggs are deposited in the water. That's what one learns in school. They become tadpoles. The tadpoles then go through the process of metamorphosis to give rise to the terrestrial form. These pictures come from Diego Gomez Hoyos. He is I do not know him. He comes from Colombia, and somehow he said he wanted to send me a poster with these images. And I said to him, please send me better an electronic version of the poster. And so I think that is very pretty. Well, think of the many frogs that I was talking that they occur in Ecuador. The point is that water comes to be a limiting resource for reproduction. Water, we are talking of the rainforest. There is a lot of water, but it comes to be a limiting resource for reproduction. In 1972, William Dullman and his student discovered in 3 square kilometers in Santa Cecilia in Amazonian Ecuador 82 different species of frogs. What is special about 82 different species? The entire continent of North America, this includes Canada, the United States, and Mexico, have a total of 82 species. So how can then frogs live in such small area with so many different species? They have evolved different reproductive modes. Early development, when the eggs are deposited in the water, it's the most dangerous time because predators are ready to eat them. What I tell my students, well, as you eat a fried egg in the morning, well, predators will take a frog egg for breakfast or lunch or whatever. And I decided to give you a picture to show you some frog reproductive modes. This picture is from my colleague, Santiago Ron. You can actually find it in the internet. Not him, the picture. This species attaches the eggs to the leaves of vegetation. Just imagine this plant above the water. Embryos, so the most dangerous, vulnerable eggs are above the water. They develop when the rain comes, then the tadpoles drop into the water. A tadpole at least can swim and can hide itself. This is another strategy to deposit in this case, on the vegetation. And what is this doing, this frog? This is some sort of parental care to avoid that other frogs or other predators may come and eat the eggs. And then again, the same thing, when it rains, the tadpoles will fall into the water. Species I show here is a Lutherodactylus coqui. This is actually a frog from Puerto Rico. We don't have a Lutherodactylus in Ecuador, but there are related genus in our country. This frog has terrestrial eggs without tadpoles. So the male has found a place where to deposit the eggs. The female deposits the eggs. There is some plexus, and then one frog is around the embryos. 30 to 40 days later, according to species, then baby frogs emerge. They don't have a tadpole stage at all. If frogs would have been studied in the tropics instead of in the temperate zones, they would say, well, the invention of the tadpole occurs only in a few species. More than 400 species of frogs have just terrestrial eggs. I now come, so I didn't tell you about the 20 different modes of frog reproduction. I now come to the first part of my talk, the biology of frogs. I divided the frogs, those that have rapid development in comparison with those that have slow development. Later in my talk, you will see what I mean by rapid and slow development. For the moment, just believe me. In this slide, I am giving you the scientific names. Well, I am going to talk about Cenopus laevis. I am going to talk about a frog that produces foam nests, and the name of this one is Engistomops randi. In the talk, I will just talk about foam nesting frog. A dendrobatid frog, and the scientific name is Colostytus machalilia, and of course, I will also mention the marsupial frog, Gastrotheca ribambe. So Cenopus and foam nesting frogs have rapid development. then robotic frogs and marsupial frogs have slow development. And then you will ask me, why do we study the development of frogs other than Cenopus? There is a historical reason. When I finished my doctorate, which I did in the United States using Cenopus as a model system, I returned to work in Ecuador. I did not have the means to buy Cenopus. I decided to go to the gardens of the university and find a frog. You know, all frogs should be the same. And I found that in the gardens at the time there was a marsupial frog. Most of my career has been done on the biology and development of marsupial frogs. And I would say when we analyze other frogs, Cenopus is a fantastic system because it gives us the grounds for comparison. And when one looks at other frogs, one can sort of compare and see, oh, this looks different. And then that would allow us, or others, to look into the development of Cenopus from another perspective and understand embryonic development better. In addition, of course, I train Ecuadorian students. Let me mention that the early development of Cenopus is better known than that of human beings. In regard to its biology, Cenopus is a special frog because the adult is an aquatic frog. The eggs of Cenopus are deposited in the water and they develop rapidly. I come now to the foam nesting frog. How is the foam nest produced? At the time of mating, that is the time of amplexus, which occurs at night. So in pools of water, the eggs are deposited with a very liquid egg jelly. The two frogs beat their legs and they produce this foam which floats above the water. And the foam nest will float for about 1 or 2 days. The tides become eggs and then later on the non-synchronous oogenesis. To our surprise, as I said, the early development was not known and we were able to produce a table of developmental stages for this frog. So here is the foam nest, which measures about 5 centimeters in diameter. The eggs are white, and here is an egg within the foam nest. The early development looks like that of albino Cenopus. The egg size is also like in Cenopus. When it gets to the tail bud embryo, then the embryo looks different from Cenopus. The production of the foam nest, as I said, is a protection against predation. These foam nests are deposited in temporary water where maybe there are less predators. Of course, they are subjected to desiccation, so development has to occur rapidly. I now come to the frogs with slow development, and I come to the dendrobatic frogs. Dendrobatic frogs are known worldwide because they are very colorful. Some of them are bright red and some of them are yellow or blue, and many amateurs keep them in captivity. And we look at the Colostetus, this dendrobatic frog, in our laboratory. The bright colors is an advertisement signal. They are quite poisonous. And the Indians, local aboriginal inhabitants from South America, used skins of these frogs to poison arrows to kill small mammals like monkeys. And that's why they are called the poison arrow frogs. The male, these frogs are territorial. The male finds a place where, which is appropriate for a nest. The eggs are terrestrial. For example, we provide them with, in the past there used to be the film boxes, which were great. And we provide them as hiding places. And so a male will attract the female. She deposits the eggs in the nest. The male knows his territory, his entire life. He's just supervising and advertising his territory against other males. because other males will come and will eat the eggs. After 19 days, so here is a nest that we isolated. After 19 days, the tadpoles hatch, and they attach to the back of the father, and then he transports them to the water. In other species, it is the mother, the one that transports the eggs to the water. Surprisingly, again, in the case of this frog, nothing was known about the early development. These drawings were produced by a former collaborator, Oscar Perez, and as I said before, the entire development takes 19 days. If you look at these images, the characteristics of the early development in the robotic frogs look like frogs. So I now come to some features of the biology of marsupial frogs. This picture in the middle shows a female marsupial frog that is incubating embryos. The female has a pouch on the back, so the name Gastrotheca is not correctly given because it is not like a kangaroo on the belly. The pouch is on the back, and it opens above the cloaca. And the question is, how did the eggs get into the pouch? That occurs at the time of mating. Mating occurs on land. The eggs come out of the cloaca of the female, and with his hind feet, the male pushes them inside the pouch. Fertilization occurs at that time. The characteristics for in vitro fertilization for this frog, I have not been able to work them out, but I can induce ovulation by the administration of chorionic gonadotropin. The female then carries the embryos on her back for about 4 months. This female may have an average of 120 eggs in her back. And I always wondered, how is it possible that these embryos develop for this very long time on the back of the mother? Well, the pouch before the incubation of embryos has a characteristic, a histological characteristic of frog skin. It looks like an enfolding of the frog skin. During incubation of embryos, it grows between each embryo, making chambers of pouch tissue for each embryo. The embryos, the gills, which are filamentous in other species, are discs that envelop the embryo, and so exchanges with the mother occur between the pouch tissue and the bell gills. At the time of birth, the female goes to the water. In this species, she holds herself with the front legs to a surface and bends the feet to distend the opening of the pouch and aid in the emergence of tadpoles. Here is the head of a tadpole that was just born. Here there is another one. Here is the tail of a third tadpole. Although this frog produces tadpoles, the majority of marsupial frogs protect their embryos until they are baby frogs. So we looked at the early development of the marsupial frogs. Luckily, one can go with a pair of forceps into the pouch and get a few embryos at a time. I have also developed a saline solution to culture these embryos. The eggs are large. In this species, they measure 3 millimeters in diameter. They are white, and to discover the features of early development is very hard. Here, this is the dorsal blastopore lip, which took us years to discover. Once the blastopore is closed, an area of small cells occurs in the area of the closed blastopore. This embryo has been stained for cell surface. This is an embryonic disc, and from this embryonic disc, the body of the embryo will develop. The gastrulation in marsupial frogs is the most divergent mode of gastrulation among frogs. And so that was a brief introduction into the biology of the frogs. And I now come to the second part of my talk, and I want to do a comparison of frog development. The embryo that I show here is a Cenopus embryo, and it is being looked from the lower part, which is called the vegetal part. You will see the formation of structure in this area. Cells come to this area and they move inside in a process called involution. I want to show you a film. Actually, this film comes from the laboratory of Jim Smith at the Gurdon Institute. Can we see the film, please? So there you see, there is the formation of the blastopore, and now you see something special. The embryo is changing shape from the round spherical shape of the embryo, then it changes into the elongated shape of the tadpole. Just to recapitulate what you just saw in the film, I want to show you again some of the images from the development in the dendrobatic frog. Cells move towards this area, which is the dorsal blastopore lip, and they move inside in a process called involution. The dorsal blastopore lip grows around, and eventually it surrounds an area that's called a yolk plaque. Involution occurs all around the blastopore. The yolk plaque becomes smaller and smaller, and at the same time, the embryo, which is spherical, will change into the elongated shape of the tadpole. We study gastrulation. Why do we study gastrulation? Because gastrulation is a common process among vertebrates, and if we learn something about frog gastrulation, it will be important for the understanding of early vertebrate development. During gastrulation, the body plan is established with the formation of the ectoderm, the mesoderm, and the endoderm, and I am using images from the book of Wolpert. During gastrulation, in the mid-gastrula, the process of dorsal convergence and extension elongates the body, and Ray Keller has done a lot of these studies. So if you take just the mesoderm, which is isolated here, in the mid-gastrula, the cells move towards the dorsal side, and at the same time, the body begins its elongation. That is called convergence and extension. So recapitulating what I am just saying, then in the mid-gastrula, the body of the embryo begins its elongation by the process of dorsal convergence and extension to form the tadpole. We now know that the planal cell polarity pathway controls convergence and extension. At the base of the planal cell polarity pathway is the gene brachyury. I mentioned brachyury because I will be talking more about this gene later. The dorsal blastopore lip, which you saw in the previous images, is important because of the work of Spemann and Mangold. They took a Euro-DL embryo which was pigmented. They isolated the dorsal blastopore lip and placed it in another embryo which was less pigmented. And the result was the duplication of the axis, and this work led to the Nobel Prize, which was awarded to Hans Spemann. By then, Mangold was dead. The organizer has different properties during development. The early blastopore lip, when transplanted, duplicates just the head. If the transplantation is done from a late embryo, from a more advanced embryo, only the trunk and the tail is duplicated. Therefore, the organizer is thought of consisting of the head organizer and the trunk organizer. The head organizer is represented by the precaudal plate, and the trunk organizer by the notochord. I will be talking about these things later. In 1991, the technique of in-situ hybridization was applied to frogs, and in the laboratory of Edith de Robertis, Ken Chow and collaborators isolated the gene cuscoid, which very interestingly was expressed in the dorsal blastopore lip, in the organizer. Additionally, they described that if this gene was misexpressed in the ventral side, it produced axis duplication, and therefore it was called an organizer gene. So for a very short while, one thought that the organizer problem was solved. A few months later, from the laboratory of Igor David, Masanori Taira and collaborators isolated another gene, the gene LIM1, which is also expressed in the dorsal blastopore lip. Its misexpression in the ventral side produces axis duplication. Right now, we know that there are a number of genes expressed in the dorsal side. There are others expressed on the ventral side, and that the gene interactions is quite complex. I want to mention that organizer genes are conserved in vertebrates. My mentioning the gene LIM1 here it comes with the fact that I will be talking about that gene. So summarizing what I have been just telling you, either transplantation of the dorsal blastopore lip to the ventral side or the misexpression of organizer genes in the ventral side produces axis duplication in Cenopus. I come now to explain you the time from fertilization to the gastrula in the frogs I was mentioning. So Cenopus and the foam nesting frog, they have eggs of 1.3 millimeters in diameter and take 14 and 24 hours for this process. We call that rapid development. The Robotic frog is slightly larger, and it takes 4 days to do that. So that's a difference between hours to days. And the Marsupial frog, for the same process, requires 14 days. The frog I study has eggs of 3 millimeters in diameter, but other marsupial frogs have eggs of up to 1 centimeter in diameter. We first wanted to know about the morphology of gastrulation. So these are sagittal sections of Cenopus and of the foam nesting frog, and the appearance is quite similar. So these are mid to late gastrula. There has been involution, and a new cavity has been formed. That cavity is called the archenteron, and the archenteron is large. Elongation of the archenteron occurs during gastrulation in these frogs. When you look at the appearance of the gastrula from the dendrobatic frogs, externally it looks like a frog. When a section through the mid-sagittal section is done, notice that the archenteron is smaller and that the blastopore lips are thick. This frog has a delayed elongation of the archenteron. The thick blastopore lips are here even after closure of the blastopore. So this area is called the circumblastoporal collar. In Cenopus, it's much smaller. In the marsupial frogs, this is a marsupial frog embryo at the time when the blastopore is closed. If I stain for cell surface this embryo, around this closed blastopore is this area of small cells, which is the embryonic disc from which the embryo will develop. If this embryo is bisected, the archenteron is tiny and the circumblastoporal collar is very large. So in conclusion, slow development of these frogs is accompanied by a delay in archenteron elongation. And here you have the aspect of that the circumblastoporal collar consists of many small cells. Compare the size of these cells with this cell in the floor of the archenteron. So these are drawings made to scale of a marsupial frog and Cenopus, and what I see is a different timing in elongation of the archenteron. Cenopus is elongating the archenteron during gastrulation. This marsupial frog is elongating the archenteron later. For our studies, we did a comparison of LIM1 and brachyury expression. The tool was immunostaining. We used polyclonal antibodies against LIM1 and against brachyury. They come from the laboratory of Masanori Taira at the University of Tokyo and Bernd Hermann, who was then at the Max Planck Institute in Tübingen. The proteins LIM1 and brachyury are transcription factors. Therefore, they accumulate in nuclei, and this gives this punctuated appearance to the immunostainings. to demonstrate that these are actually nuclei. This embryo was double stained for DNA, and one can see that the brachyury immunostaining quenched the DNA staining. Let me remind you about the expression of brachyury in Cenopus laevis. These images come from Wolpert. In the early gastrula, brachyury is an early marker of mesoderm induction, and it marks the presumptive mesoderm around the yolk plaque. Starting in the mid-gastrula, brachyury is in addition expressed in the notochord. So it is expressed in the presumptive mesoderm and in the notochord. Expression of brachyury in the presumptive in the notochord is evidence of dorsal convergence and extension. In both Cenopus and in the foam nesting frog, elongation of the notochord begins in the mid-gastrula. Therefore, in both species, convergence and extension and elongation of the notochord start during gastrulation. I can say that gastrulation and convergence and extension are linked in the rapidly developing frogs. This means that there is elongation of the body during gastrulation. As I showed you earlier, during gastrulation, the process of convergence and extension begins to rapidly produce a tadpole. How is the situation in the slow developing frogs? And here I show you images of brachyury expression in marsupial frogs. There is expression in surface cells that follow the blastopore lip in marsupial frogs. And in the early gastrula, only these cells had been detected. Once the blastopore is closed, a deep expression comes in the closed blastopore area and in the notochord. And it's expressed then in the notochord and tail bud. The same occurs in the dendrobatid frog. So this is a late gastrula. You don't see any notochord at all. Once the blastopore is closed, then brachyury gets expressed in the notochord. So what I am showing you is that in slow developing frogs, convergence and extension and elongation of the body occur after gastrulation. And here is a comparison of the expression of brachyury in the various frogs. I forgot to mention that in Cenopus, the expression of brachyury is an internal ring. There is an internal ring that shows the prospective mesoderm. Starting in the mid-gastrula, then the notochord can be detected. In the frogs with slow development, the notochord can be detected once the blastopore is closed. It's unknown whether Cenopus has this surface expression of brachyury. The implications are as follows. In Cenopus, you get involution at the blastopore, elongation of the archenteron, and notochord elongation with convergence and extension, all of that starting in the mid-gastrula. Whereas in slow developing frogs, the elongation of the notochord and convergence and extension has been delayed until after blastopore closure. This indicates that frog gastrulation is modular. And second, it indicates that elongation of the body, that is convergence and extension, is not necessary for gastrulation. So summarizing what I am trying to show you, I would like to say that there are different ways to make a frog. You can either do simultaneously gastrulation and convergence and extension, and you produce a frog, and that occurs in the rapidly developing frogs. Or gastrulation can be completed, and then convergence and extension occurs, and that is the characteristic of slow developing frogs. But it is also a characteristic of the coquille, and the coquille is one of the most rapidly developing frogs that exists, because from egg to frog, it takes about 40 days. So what I would like to say then, it is a recurrent fact that sometimes there is simultaneous occurrence of gastrulation and convergence and extension, or there is separation of this process. I mentioned that I will talk about limb 1. These are embryos of Cenopus immune stained for the protein limb 1 to give you an information about the expression pattern. Limb 1 has a complex expression pattern. In the early gastrula, it marks the dorsal blastopore lip. Slightly later, it marks the entire prospective mesoderm. But then that expression gets restricted to the notochord and precaudal plate, so it is a marker of the dorsal mesoderm. Later, it is expressed in the pronephros and in certain cells of the central nervous system. This expression pattern occurs in all of the other frogs that we analyzed, so it is highly conserved. And briefly I show you, then here it's expression in the pronephros of all of the analyzed frogs and in cells of the central nervous system of all of the analyzed frogs. What about the dorsal blastopore lip? Well, very nicely, the dorsal blastopore lip in all of the frogs expresses limb 1, so the expression of limb 1 in the dorsal blastopore lip is conserved. It is very nice that we have two markers of the prospective mesoderm. In Cenopus, brachyury is the marker of the prospective mesoderm, but for a short time, limb 1 is also expressed in the prospective mesoderm. In the dendrobatid and marsupial frogs, I showed you that the limb 1 expression is delayed to the post-gastrula, but limb 1 is expressed at this time, so this means that there has been mesoderm induction. It is only that the process of dorsal convergence and extension has been delayed, and brachyury is within that pathway. In rapidly developing frogs, limb 1 is expressed in the notochord and precaudal plate, both in Cenopus and in the foam nesting frog. The notochord and precaudal plate are limb 1 positive in the mid-gastrula. What about these other frogs? This one does not elongate the notochord during gastrulation, but the precaudal plate is limb 1 positive. Here the blastopore is closed, and this is the area of the precaudal plate, and here I show you the sections. So only the precaudal plate was detected during gastrulation. The same happened in the marsupial frog. Once the blastopore is closed, then limb 1 marks the notochord. So this result of the notochord comes along very nicely with the previous brachyury expression. So here is a summary slide. The precaudal plate and the notochord develop simultaneously in the midgastrule of Cenopus, and the development of these structures is separated in time in slow developing frogs. The implication of the comparison is that the head and trunk organizers are separable from each other. It also implies that the different ways to make a frog imply a different timing of gene expression in the organizer. I want to conclude this talk by thanking my present and past collaborators. Here is Natalia Sáenz, who has a poster. I invite you to visit her poster. I also want to thank the many international colleagues that discuss with me the ideas and allow me to go ahead in my work. And I thank you very much for your attention.
Questions? Okay. Until you will get, I will ask one question. So I have one, and then Ariel.
Yes.
Can you comment on any correlation between changes in developmental timing and ecology, the ecology of the frog?
Well, I do not know much about the ecology of the frogs, but I am trying to say that the there is a correlation between the adaptations, making development slower or faster, and either placing the eggs in the water or finding ways to protect the early development. So if a frog protects the early development by placing it in the pouch of the mother or by placing them in terrestrial nests, then they can uncouple gastrulation from convergence and extension.
My question is actually related to Ramit. It's more of a comment than a question. I think that one thing that you didn't really talk about is the effect of the size of the egg, and specifically the amount of yolk in the egg, and how that affects development. And I think that in many cases, the slow developing frogs are frogs that, as you say, are protected. If the egg is protected, then it can afford to develop more slowly. If it develops more slowly, it needs more yolk to feed the embryo during development. And because it has more yolk, this has all kinds of effects on the way development can proceed, the way gastrulation can proceed, etc. And an example is, for example, Hyperoleus, which has a relatively small egg, but a lot of relatively small egg, relatively fast development, but a lot of yolk, and it develops very similarly to coquille or Gastrulation.
Your comment is very nice, but the picture is more complicated. I always thought that if the eggs were larger, contain more yolk, develop more slowly. No, they can be very large and develop very fast. The best comment is Gastrulation riobambe and Eleutherodactylus coquille. Coquille has eggs of 3.5 millimeters in diameter. Gastrulation 2 to 3 millimeters in diameter, and the coquille develops as fast as Cenopus. So there is no problem in dividing a large egg.
But it's not so much the speed, but the way development proceeds. Because there are different things that are affected by how much yolk there is, so different phases in development can become uncoupled.
Yes, I think you might be right. Yeah, I think Val has a question.
I have a biological question. You discussed 20 different reproductive modes in Ecuador. Can you find many of them in one tree? Several different modes in one jungle tree?
So your question is, if I go and visit a tree, and a botanical tree, and whether in this plant there are different frogs with different reproductive modes, I imagine that yes. I haven't done it, but I imagine that yes.
So, one moment.
So, what are the phylogenetic separation times for the species you were talking about?
Excuse me?
What are the phylogenetic separation times for the species that you were comparing?
I don't have that data in my mind. I know they belong in separate families, different groups. You know, we're talking of Cenopus, and the marsupial frogs until very recently were considered hylid frogs. So the frogs I am talking, they are modern frogs in Cenopus. Mm-hmm. You have one question up there.
Okay.
There is one
Over there.
Oh, there is another.
In the slow developing species, so after cells involute, there is a phase when they just sit there and wait adjacent to the organizer before they start elongate, right? So
Maybe
does that
they
mean
don't
they're
sit,
exposed?
Maybe they are doing things that I do not know.
Right, but does that mean they're exposed longer to signals from the organizer? And does that affect the body morphology or proportions? Or is the signals also weaker or begins later?
That is the questions I would like to get a hand into. Yes, I do not know.
Okay, Dave.
And this person over there has a question.
Hola, mucho gusto. Hi, over here.
Yeah.
How does the fact that the steps which are considered to occur simultaneously in Cenopus show or give us any insight about the fact that they happen sequentially in marsupials? Does that give us any new insights as to development of vertebrates or frogs specifically?
I was thinking of the talk of you, Mark, today, and that this is an example where you, in nature, these are two processes. We think that convergence and extension is part of gastrulation, but it just happens that we looked at only one animal. These are two processes, two different processes. So that is why at the end I said the organizer gene expression may vary in these other frogs. I don't know if my comment answers your question.
Yeah, this is probably an easier question to ask than for you to do the experiment. Have you been able, do you have the tools available to look at, I'm over here.
Yeah.
Yeah, okay. Do you have the tools available to, for instance, look at a target of planar cell polarity like Wnt11 and to actually see this, the great difference that you would expect to see, right, where they probably would be different time frames of expression, maybe different positions, which would give a hint as to why, that the actual driving force of why the conversion extension is different?
I would say I don't have much of the tools.
Yeah, okay.
There are several problems associated. First of all, the technological aspects, and second of all, the collaboration of the frogs. So, you know, if you have Cenopus as a well-established system in which you can inject and you come in the morning and you have the eggs, when you depend on natural matings, that is a problem. The eggs of the marsupial frog, you puncture them and they are like under pressure. The yolk comes out. So I don't have the skills in part.
You mentioned that the head and trunk organizer are separated. Can you speculate why it is? I would think that they have to function somehow in coordination, and also in Cenopus, I think there is some coordination between the two organizers. Can you speculate? And also, can you do the Spearman experiments and see that they actually give rise to two different things?
That is an idea that we keep in mind. Is it worth to do the Spearman organizer experiments into these frogs or not? So I think if the work of Spearman would have been done in these frogs, he would have had just the development of the head, like I showed you that in Cenopus or in amphibians, the early blastopore lip just produces the head, it does not produce the trunk. So in a way, that head and trunk organizers are separate. It is also shown in that kind of amphibian experiments. I think that's what I want to say. Thank you. Yes, he has a question over there. He had been asking forever.
Yes. Last question.
Thanks for a fascinating talk. But I would wonder if you have, even as a speculation, any idea, or how would you like to tackle the problem of how one developmental pattern is transformed into another? I mean, if there is like the evolution of of one developmental system to another. And I'm thinking about people who started to talk about developmental rates, like Gavin De Beer and Richard Goldschmidt. They were always looking at how one pattern would develop into another. And I found it that you didn't really touch on it, and whether you'd like to go into it in the future, or you have any ideas concerning this transition.
Well, it is very interesting to think about how one development pattern developed into another. If you think the marsupial frog has actually a parallel line to the terrestrial vertebrates, so that they are in a way inventing a new development pattern, and that implied the separation of the head and trunk organizers. So far, that is the only thing that I have found.
Thank you.
Thank you.
Okay. Thank you very much.